So there are animals that can regenerate complex body parts, such as the nervous system, remarkably well. So there is, for example, salamanders or fish that when they lose a part of their body, such as their tails, they can regrow it in just a matter of weeks. But unfortunately, when our brain and spinal cord gets damaged, we cannot regenerate spontaneously.
In most tissues in our bodies, such as the skin, the skeleton or the gut, cells regenerate very well. Some of them even get renewed every couple of days. When one of these tissue types get damaged, populations of stem cells get activated and generate the cells that have died after that injury. In the brain and the spinal cord, there are stem cells or progenitors that also have some capabilities to create new cells, and in fact, some of them do. But the majority of the stem cells cannot produce new nerve cells, as they don't have all the equipment they need to replace the cells that have been lost after an injury. That's what Enric Llorens Bobadilla and his research team at Karolinska Institutet are trying to solve.
So the stem cells exist and they have all they need to respond to an injury, but what they do is form a scar instead of forming the tissue that you would need to repair the damage. We're trying to convince the brain and the spinal cord to repair themselves. So we're trying to achieve that they gain the regenerative abilities that unfortunately evolution did not provide us with.
The first step for the researchers is to understand how neurons are formed in embryos. They therefore analyze the instructions that the stem cell reads from a DNA and follows all the steps they go through to make a particular neuron type. When the researchers understand how the embryo does this, they might be able to recreate the process in an adult.
So now for the first time we can understand all the genes that are active when a particular type of neuron is born in an embryo. And in addition to that, there are now tools such as CRISPR that was recently developed that enable us to activate specific genes in specific cells. So for example, in our case, when we want to activate a specific gene that has been active in embryonic stages to make a specific neuron type, but is no longer active, we can use CRISPR in this adult tissue as a tool to switch on and off the genes that we want in the cell that we want. And this is something that was simply not possible just a couple of years ago. So what we do is design in the lab these pieces of DNA that carry the information to bring the modified CRISPR, so basically the on switch for a gene, and then we package it into modified viruses that are non-pathogenic, so they don't cause any disease, so they are very safe, and then deliver that virus to the organism. And then once that virus enters the organism, it carries all these CRISPR tools to specific cells, and in that cell, this would activate the specific genes that we aim to activate using that CRISPR tool. And then we can test whether the activation of that gene led to a different behavior in that cell. So was the cell able to generate a neuron, yes or no? If yes, then we test it further. If no, we try a new one. Are there more in that area? So this was the scar-forming cell. If what we deliver with the virus, which in this case make it be green, works, then this cell will convert into a cell that expresses the neuronal gene, which is here depicted in magenta. In addition to all this, now we also have the computational tools, so all the bioinformatics analysis, machine learning methods that allow us to try to understand these large data sets and prioritize which are the key genes controlling such specific cellular behaviors. So this is one of the main aspects. So we don't need to test 20,000 genes. We can predict which are the ones that are likely to be most important for the process we are studying.
If scientists can get the body to regenerate nerve cells, it will open up entirely new possibilities for healing brain and spinal cord injuries and curing various degenerative nerve diseases such as Alzheimer's, Parkinson's, and ALS. But there are many types of nerve cells that must be regenerated to repair nerve damage, and every time Enric Llorens Bobadilla and his colleagues succeed in reprogramming, we get closer to the solution of healing nerve damage.
When a student sees something in the microscope that, you know, it's so exciting that there's nothing that can that is better than that moment. Because then when you see that all these years of work suddenly materialize into something potentially useful, and there's nothing that is as good as that moment. So yeah, that we're always looking forward to getting more of those.